In this video, we're just going to go through 3.3 photosynthesis, which I believe is the week 6 work we've been doing this week. So in the booklet, you'll notice that on every topic, every page, there's a visual representation, either as a picture or as a summary diagram. There's also the written text, which should help us to learn the information. So the general word equation for photosynthesis is carbon dioxide plus water in the presence of light gives us sugar in the form of glucose and oxygen. When you were learning about respiration, potentially in third year, you might have learned go cowie. So glucose plus oxygen gives carbon dioxide plus water and energy. In photosynthesis, it's actually the opposite. So instead of go cowie, it's cow we go. So CO2 plus water, cow, in the presence of energy, that's the E, so COW, cow we, and then glucose and oxygen, go. So if you're struggling to remember photosynthesis, it's cow we go, and if you're struggling to remember respiration, it's go cow we. So the raw material consists of carbon dioxide from the air, water from the soil, and these are combined using light energy from the sun, which is trapped in chlorophyll to produce sugar in the form of glucose and oxygen. Really, in the National 5 course, we want to use the term glucose rather than sugar. Photosynthesis is enzyme controlled. So when we look a little bit at the enzymes topic in Unit 1, you'll see what that means. But essentially, all of photosynthesis is controlled by enzymes. In the first stage, it's dependent on the light energy. So this is what we call the light reactions. So if you're asked for the name of that first stage in photosynthesis, it's light reactions. So the energy from the sun is trapped in chlorophyll in the leaves of green plants. It can also be in the stem to some extent, but anyway. The light energy is converted into chemical energy. Some of this is used to split the water molecule into hydrogen and oxygen. The hydrogen is passed into the second stage of photosynthesis, and the oxygen just diffuses out. So the water is split, the oxygen is treated as waste product and is allowed to leave through the pores of the leaf, but the hydrogen is kept and used on to the second round of photosynthesis. ATP is also produced. This is an energy molecule which, again, we will use in stage 2. So as I said, this is the text, this is the visual version. So light energy goes into chlorophyll. A lot of people get confused and put water in here, but water is already in our diagram. So light energy goes into chlorophyll and is used to make chemical energy, as well as splitting water into hydrogen, which is passed into the second stage, and oxygen, which is passed into the air. In the second stage, so we have the hydrogen and the ATP from the first stage, it carries forward. So if we look at the summary first, if you try and spell hydrogen correctly with an O, that's always useful. So the hydrogen and ATP from the previous stage are brought on. The hydrogen is used to join several carbon dioxide molecules into a ring, which makes glucose. So in the text, the second stage is enzyme controlled. This makes it also temperature controlled because enzymes only like particular temperatures, as we'll find out in a couple of minutes. Hydrogen from the first stage is combined with CO2, carbon dioxide, to make sugar, which again we should call glucose. The energy required for this comes from ATP, which we got from the first stage. That's one of the things the light energy was used for. This is quite a cute little summary diagram. So it shows for stage 1 and stage 2 photosynthesis, what the name for the stage is, what is required, what the energy source is, and what is produced. So in stage 1, the light reactions, light is our energy source, and it's used to split water into hydrogen and oxygen, but light energy is also used to make ATP from something called ADP and inorganic phosphate, which we'll talk about later. In the second stage, carbon fixation, the energy is from the ATP produced in that first stage. It joins the hydrogen, again produced in the first stage, and CO2 from the air to make glucose. So you might be thinking, well, actually, it looks like light and temperature and CO2 are going to be quite important in this reaction. We'll talk about that. So that sugar that is used can be made into several things. It can be used in respiration. It can be converted into useful substances such as starch and cellulose. So you need to know the three general fates of glucose. It can be used in respiration. It can be kind of modified to make cellulose for cell walls. So you know from third year and from Unit 1 of National 5, possibly, that cellulose is what makes plant cell walls. And starch is like plant fat. It's their storage molecule for glucose. So I talked about these three things being quite important for where are we? Photosynthesis. That's where we are, for photosynthesis. We tend to call these the limiting factors. Some textbooks will put water in as a fourth. We don't tend to do that because it's a slightly different, when you look at the chemistry of it, it's a slightly different thing. So temperature, carbon dioxide, and light intensity. These are our limiting factors. When we talk about this, we have to say temperature, carbon dioxide concentration, because the SQA are a little bit pedantic. If you say carbon dioxide is a limiting factor, they might say, oh, do you mean the color of the carbon dioxide, or the temperature of the carbon dioxide, or whether the carbon dioxide is, there or whether it's not there? You have to talk about the concentration. How much carbon dioxide do you have? And again, you can't just talk about light as the limiting factor. It has to be light intensity. So how strong is the light? How much light is there? So we start with that light intensity. Without light, plants can't photosynthesize very quickly because that first stage is light dependent. If you take away light, they can only use what they already have from that first stage. If you increase the light intensity, the rate of photosynthesis will increase also. So here, we tend to say that, we'll talk about this anyway. The limiting factor at point 1 is light intensity. So my two rules are, if the line is going up or going down, then whatever's on the x-axis is your limiting factor. So the line here is going up, which means it doesn't matter what else is on the graph. Light intensity, what's written there, is limiting factor. If that was carbon dioxide concentration, it would be the limiting factor. If that was temperature, it would be the limiting factor. If it was number of bumblebees, it doesn't matter. What this shows is that as you move along the x-axis for number of bumblebees, it looks at the rate of photosynthesis, for some reason goes up. Here, what we can say is of our three limiting factors, it looks like if you increase light intensity, so 5 units, 6 units, 7, 8, 9, 10 units of light, doesn't seem to do much to the rate of photosynthesis. So all we know at this stage is that light intensity is not the limiting factor. It's either going to be the temperature or the concentration of carbon dioxide. And similarly here, as I said, rule 1 is that the line is going up, so the limiting factor is CO2 concentration. And all we know for sure is that carbon dioxide is not the limiting factor at point 2. So it's either light intensity or temperature. Temperature is a bit different. If it's too cold, photosynthesis won't happen because there's not enough energy. And if it's too hot, the proteins get what we call denatured. Now, this again is a term from Unit 1 of the course, key area, I want to say 2? No, 5. 4, key area 4. Proteins. What it means is that the shape of the protein is no longer close enough to the shape of the thing that it's trying to work on. So the enzyme doesn't work very well anymore. So this is an odd case where when the line goes up and when the line comes down, that temperature is the limiting factor. Or temperatures, if you want to say. Interestingly, the only time in this graph that temperature is not limiting factor is right at the very middle there. That is a point where temperature is no longer controlling the rate of the reaction. Something else, light intensity or carbon dioxide concentration is. So plant growth is limited by how much sugar we have. How much sugar we have for respiration. And the greater the respiration, the greater the growth, and you get more sugar produced. One thing I just wanted to point out, so we said here it's either light intensity or temperature. If we had a graph like this, if we ignore the top two lines, if we just take the bottom one at the moment, so the bottom one, we are increasing light intensity at 20 degrees Celsius and with 0.1% carbon dioxide. Don't worry about the units, degrees and percents, but you can see that over time here, limiting factor is light intensity. And then at this point onwards, all the way along this line, it's either temperature or carbon dioxide concentration. So previous questions, we've had to say, well, we don't know which one it is. But actually, if at this point here, you increase the temperature, so you change it from 20 degrees, say, if we make up an arbitrary light intensity, let's say it goes from 0 to 10 in light intensity, that's light intensity 5. At light intensity 5, if we raise the temperature by 10 degrees, but keep the light intensity the same and the CO2 concentration the same, what happens? So what we find is that the rate of photosynthesis goes up. So at this point, the temperature could be, if we ignore the third and the fourth line, the temperature is a limiting factor. Because we know if we keep light intensity the same and CO2 the same, the rate of photosynthesis will go up. So if we're now on this line, if we say, well, actually, if instead of increasing the temperature, we kept the temperature the same, but increased the concentration of CO2, what would have happened? And actually what you find is at that point, unfortunately, both temperature and CO2 became limiting. So if you either increase the temperature or the concentration of carbon dioxide, it makes a difference. And again, at this point here, if we think, well, what is the limiting factor? If you increase the temperature, it apparently was a limiting factor because the rate of photosynthesis goes up. We don't know if CO2 is a limiting factor. It could be that, say, at this point here, 10 light intensity units. At 20 degrees, if you increase the CO2 concentration to 0.3, it could be that the line it stays where it is here. So it could be that at 20 degrees, 10 units of light, 0.3% carbon dioxide, you get the same result. So we don't know. Sometimes you will get a graph like this, or probably a more simple one, in your exam. It's very unlikely, but we like to teach these kind of things just in case it comes up. Hopefully that made some kind of sense. If there's anything in particular you don't understand, please ask.